Welcome everybody to the RACI Bioactive Discovery and Development Group events. This is our second for the year. And of course, this is the second online event that we've ever hosted, but our last one was quite successful, I think. We had Brett Carter from the CRC for Cancer Therapeutics, or the ex-CEO of that. And I think everyone who attended that seemed to enjoy that that event, and we hope today is just as big a success. And I really do thank everyone for making the effort to join tonight. It's a very good show. I think we're expecting over 60 people, so it's really a nice time to get together as best we can. Normally we would be at a pub, so there's normally an event that we hold every quarter where we do have a speaker and a dinner and some pre-dinner drinks and canapes. So I would like you all to just encompass that feel, grab a drink and sit back. It should be a nice, entertaining presentation. So for those of you who've not seen our group or don't know about us, I just would also welcome you and thank you for joining. We are a group which is a specialist group of the Victorian branch of the RACI, the RACI being the Royal Australian Chemical Institute. So obviously a professional group that's core, I guess, is medicinal chemistry, but we do encourage attendance and participation from all disciplines and anyone interested, really. So it's a very active group. We do try and keep things happening and discussing events, topics, and so tonight is certainly part of that. So just a bit of housekeeping before we do start. The group, obviously, we've got online for the foreseeable future. I think this is going to be the forum or the format for the remainder of the year. We do aim to hold an event every month, so please stay tuned for the next one, and hopefully you can attend that. I do thank the committee for helping get this together again, and I certainly would like to thank the sponsors again. So Davies, Collis and Cave, DCC patent attorneys, again have sponsored us. They're a long-term sponsor. And 360 Biolabs, they're also a very key sponsor, and I think specifically for tonight we've been able to keep prices low. So it's obviously a challenging time for the RACI, for all of for everyone really, to keep activities and operations going and not going out of business or losing too much money, and so we've managed to keep that price low. So thank you again. Thanks to the committee again, Rowan, Seb, Peter, and we are the committee for the rest of the year. We'll hold our AGM in December. We'll keep going as we were. I'd also like to thank Elise from the RACI. I know there have been some challenges in the registrations, so thanks everyone for persisting with getting registered. They are also facing some big challenges at the moment, so Elise has been fielding a lot of that work herself. So they're all working from home, and we will try and fix a few of the teething issues, and the next one will be even better. So just the way tonight will run, when I finish talking, which will be very soon, we'll have a presentation, and I invite people to stay on for Q&A afterwards, assuming, Simon, that you're available. Even if you're not, I think we might stay around for a chat. But again, really, it's a very interesting topic. I think everyone has a few questions. So please stay on. I'd invite people to share their video screen so we know who's talking. But look, we are on a meeting format. We're pushing the boundaries of Zoom meeting. So, but really, it's nice to see people as well. Now, moving right along, I'd like to introduce Simon. it's really gives me great pleasure to have Simon Tucker here to join us and present. When I first kind of noticed this COVID thing, you know, it all hit us at some stage that this was really serious. I thought of Simon and I thought, gee, he'd know about this, and I wonder what he's thinking. And I guess soon after that, we all thought, well, he'd be a pretty good speaker. And I do think he's probably one of the few people that can actually say, I saw this coming, or I told you so, because, you know, a lot of his work has been in pandemic preparedness. So it's a kind of an interesting time, we all know. But Simon has a wealth of pharma R&D experience, and originally from the UK, he's moved to the US quite early on in his career and spent some time at GD Searle, working on HIV and flu. And I think, interestingly, I didn't know this, but he was one of the team that developed antivirals, which is a protease inhibitor for HIV. He moved back to the UK to Glasgow University, working in gene therapy. But I think most of this audience know Simon or have heard of Simon through his time at Biota. And, you know, it's a pleasure of mine that I was part of his team for a few years. And a number of people I know tonight in the audience or joining also were part of that team. So, of course, that was a long stint, 97 to 2015, I think that was what I found out. So that's a number of years, so I worked it out. Yeah, it's 18 years, is it? So, so certainly many projects there on flu, RSV and HCV. They were some of the headline ones, but a number of smaller ones and certainly some pitches for other ones, I'm sure. So after Biota in 2015, Simon moved on and he was one of the co-founders of one of our sponsors. So 360 Biolabs was co-founded by Simon. He's currently chairman of Fertility Jumpstart and is CSO at Fibric, another local biotech company. And certainly holds a wealth of knowledge on viruses and pandemics. So I won't dwell any further, but I'd like everyone please to welcome Simon Tucker with 100 years of pandemic diseases and the progress of anti-infective drugs and other inventions. So thanks, Simon, and please share your screen so we can enjoy the presentation. Thanks.
Thank you, Ned. I now feel really old.
Let me see if I can do this. Does everyone see that? All good?
Looks good to me.
Yep,
oh,
looks good.
Please holler if it doesn't work. It's working off a laptop here and it may not work perfectly. So before I start, just apologies because I start with a deficiency in this group. I'm not a chemist. So be kind to me. I'm a virologist by training. And Ned gave me a very generous introduction there regarding preparedness for pandemics and so forth. I think there's fairly good evidence we see around us that preparedness and pandemics are two words that are very difficult to put together. And you kind of have to react, despite what preparedness you put in place, you have to react to the conditions that appear. But I thought what I'd do is I'd run through a bit of an overview on what a pandemic is, why it happens, how it gets declared, and so forth, and then run through a few examples and then end up with COVID-19 and discuss some antivirals and so forth along the way. So I'd start with what is a pandemic and why does it happen? A few ideas about interventions, what happened through history, and these are various. So you can think about societal interventions, you can also think about therapeutic interventions and so forth. So there's a whole variety of different ways we can intervene in an infectious disease. And then I'd like to talk about what's going on today, the guidance, what I've characterized as misguidance, but we can talk about that as we get into it. It's a bit controversial. What our learnings are, what our science and medicine is telling us, and what we're doing, and what's next. So I hope that feedback's not coming from me. So let's talk a little bit about epidemics and pandemics. This is something that it gets confused terribly. So what's the difference between an epidemic and a pandemic? Well, basically a pandemic is a big epidemic. It's a bit of a blurry line, but the definition is it's an epidemic that occurs across the world, across countries, and usually affects a lot of people. Generally, you don't get a pandemic announced unless it is perceived to be at high risk of widespread and substantial impact. And the perception there is actually pretty important. Just by definition, it tends to be a new pathogen, generally a virus, generally. And the reason new is important is because there's no pre-existing immunity in the population, so it can transmit readily. Now, a good example of a virus which did not become a pandemic but is new would be the original SARS, because it didn't transmit very well. It gave everyone a big scare, but it didn't become a pandemic. And often, but not necessarily always, but all the examples I'm going to talk about, it's a virus or pathogen introduced from an animal reservoir, what's called a zoonosis or a zoonotic disease. And that will become important in a minute. Okay, so here's an overview of pandemics, and the orange line is supposed to depict pandemics of the last 100 years. But basically, it picks up from the Spanish influenza, what's called the Spanish influenza. The names here are not ideal. These are names that are used in history. Spanish flu was used because a lot of the early reports were from Spain. Spain wasn't involved in the First World War. Some of the news was coming out of Spain before it was coming out of other places, and so forth. Similarly with the Russian flu, the Asian flu, and the Hong Kong flu, for instance. Don't place a lot of emphasis on the names, but they're there from history. The size of the descriptive little virus there, it represents the number of deaths, and the numbers associated with the various diseases in red show the deaths, and other colors show the deaths. So you can see with Spanish flu, for instance, we're talking about a very large number of people that died. COVID-19 is down the bottom there, 700,000 as of the beginning of August. It's approaching 800,000 now. And then various other numbers there. For instance, SARS killed 770 people. Ebola, 11.3 thousand, for instance. So quite a range of impacts from a mortality perspective, and quite a range of impacts from a disease and economy perspective. And we'll talk about that in a second. And I'm going to pick off a few of these pandemics to talk about. Now, in terms of economic impact, this is an interesting way of looking at it. These are relatively small epidemics, and only one pandemic, really, which was the H1N1, 2009. Yet you can see the numbers are very large indeed. These are billions on the left-hand side there. The number of people that have died in the SARS-CoV-2 current pandemic is substantially larger, and the economic impact is going to be measured in the trillions. So, you know, order of magnitudes off this scale, which is a really interesting way of looking at it. Okay. Okay, so let's talk about options for intervention, and we'll get into antivirals and compounds in a minute. But, you know, first of all, let's talk about the non-specific things that we can do, societal things that people can do. And so these are things like wearing masks, social distancing, travel bans, etc., all of which we're experiencing in Victoria at the moment. And I have to say it's not very pleasant. There's a very important role of diagnostics that people don't talk about. It's very important in the face of an infectious disease to be able to diagnose that infectious disease properly and track the movement of that infectious disease. And that can be in real time, as in identifying people that have the disease, and retrospectively, identifying people that have had the disease and may be immune. And this is an area that's really evolving as we speak in the current pandemic. Possible interventions, we've heard a lot about vaccines. I'm happy to talk about vaccines. I didn't put a lot about vaccines in this talk. They vary quite considerably in their availability. So if we're talking about something like an influenza pandemic, a vaccine can be available in, say, 6 months, that sort of time frame, if all the stops are taken out. If we're talking about an Ebola epidemic, until a few years ago, there was no vaccine. And obviously there is no vaccine at the moment for SARS-CoV-2. And then if we look at some of the other epidemics and pandemics, like HIV, for instance, despite a huge amount of work in trying to get a vaccine, there is no vaccine. And that could be that in some cases, these pathogens are not amenable to vaccines, or we simply haven't cracked it yet. In the case of HIV, that's probably the case. And then we talk about drugs, and there's a whole bunch of different ways of characterizing drugs. I've done it 3 ways. One is non-specific or directed at concomitant indications. So if we take influenza, for instance, some of the disease associated with influenza and the mortality associated with influenza is a consequence of bacterial co-infection or subsequent infection. So antibiotics have a role. Antibiotics have no role in a viral infection, a primary viral pneumonia, but they can have a role in secondary bacterial pneumonia. And then there's agents that have a role because they're affecting aspects of, for instance, the immune response. So dexamethasone, for instance, where a runaway primary immune response can actually be a problem. And so you want to tone it down a bit. And then we talk about specific and what I've called potentially broad spectrum antivirals. And these would be things where they work directly on the virus. And for instance, if we take nucleosides and nucleotides, because they work on nucleic acid reproduction, they have the potential for being broad spectrum. And then there's specific and narrow spectrum agents. So agents which work on specific targets, which tend to be specific for species of viruses. A good example of that would be the influenza neuraminidase. So neuraminidase inhibitors, for instance. You don't expect those to be effective in viruses that don't have an equivalent enzyme, for instance. And then I want to spend a little bit of time on medical practice because that has actually a really significant impact, and it's kind of underappreciated. We'll get to that in a minute. Okay, for some reason my screen is not advancing. Aha, here we go. So 2 slides, and I don't expect you to read this one. This is the WHO's rather detailed action plan and description of pandemic phases. It's shown pictorial in the next slide, a little bit more clearer. But suffice it to say that the WHO has a series of phases defined by the likelihood of the outbreak moving to the next stage, if you like. And that's better depicted here. And you can see that we're in the stage on the top left side, which is the human-to-human spread is widespread, and it's outside of more than one country. In other words, a pandemic. We move very fast. When I say we, I mean the world moved very fast from the early stages through to stage 6 in this particular case. And just a few words on a vaccine before moving on. So I'm not going to spend a lot of time on this. On the right-hand side of this slide, you've got a screenshot of the history of vaccines, a sort of potted history of vaccines. And you can see they start a long time ago, back in the 1800s with Edward Jenner. And that's the original smallpox vaccine, which then evolved into the vaccinia vaccine, which was used to eradicate smallpox. So it's an effective vaccine. It's not terribly safe, and it is a live vaccine, but it was used very widely indeed. And then you can see all the subsequent vaccines and when they were introduced. You can see some of these are quite early, but I'm saying they're quite early from a virologist's perspective, and I'll explain what I mean by that in a moment. If we think about what's happening today, there's a whole bunch of different ways of looking at the vaccines that are under development. The way I like to categorize them to begin with is into two buckets. One is old technology, one is new technology. What I mean by old technology is things that are tried and true, like killing the virus, inactivating the virus, and injecting it. There are several vaccines in production and in clinical trials at the moment from China, for instance, that use that approach. There are also old vaccines such as the old polio vaccine, which is basically a virus which is attenuated, but so it's live, but it doesn't produce the disease normally, but gives a very good immune response. And there is work underway and clinical trials underway with attenuated coronavirus vaccines. And then there's the new stuff, and the new stuff is generally what's being investigated in out of the Western countries. And these are things like vector mediated, where you use another virus to introduce the antigen, such as an adenovirus, or subunits, you express a piece of the virus. And then the really new stuff, which is nucleic acid stuff. All of these are really experimental. There are adenovirus vaccines used in the veterinary space, and it's been used in, adenovirus vectors have been used in gene therapy in humans, but apart from that, and some subunit vaccines, the others are very new indeed. And as with all things, a vaccine has risks and it has benefits. If we look back at the old smallpox vaccine, for instance, to give you an idea of what I mean by that, smallpox is now eradicated. So there's no benefit to having the smallpox vaccine. So it's all risk. Whereas if you have the influenza vaccine, for instance, that's going to provide a benefit because influenza recurs on a yearly basis. So there is a risk benefit that's in favor of its use. Now, I said that a lot of those dates seem to go back away, but from a virologist perspective, virology is a pretty young science. So this is just an idea of when viruses were originally described. And so you can see that a lot of these viruses are very young indeed. 1981, for instance, for HIV, and a lot of them came with Nobel Prizes. Despite that, there's a lot of antiviral drugs that have been developed and placed on the market. And here are some up to 2017. There would be a few more if we kept that figure going to the right-hand side. And you can see that principally they're in the area of HIV and hepatitis, although there are quite a lot of herpes virus drugs, some hepatitis B drugs, and influenza. Now, I'm going to pick on 3 viruses and talk about 3 viruses and their role in pandemics and the drugs that can be used to intervene. And these might be existing drugs or future drugs. So let's start off with the old classic, influenza. And you'll see a lot of people comparing the current pandemic with old influenza pandemics, in particular the 1918 pandemic. And there's a reason for that. It was huge. It killed at least 40 to 50 million people. Some estimates are larger than that. More people died as a consequence of the 1918 influenza than died in the First World War, if that puts it into perspective. The virus is a particularly nasty little fella. It's quite, if you like, mutable. It comes from principally an animal reservoir, but has variants that are highly adapted and readily grow in people, which is why we see every year an influenza epidemic. And all it takes is for the ability to grow readily in people and the newness of an antigen from an animal reservoir to get combined, and then suddenly you have a pandemic because people are not immune. And that's actually what happened back in 1918. It's also what happened in 2009, and we'll talk a little bit more about that. Now, nice thing about influenza, that we do have vaccines and we do have antivirals. So we can do something about it. Now, this is a very complicated figure. I'm not going to go through it in detail, but it's supposed to illustrate some of the things that have happened in the influenza timeline. And what we've got at the top are the various pandemics, influenza pandemics. And I've contrasted that in the top right-hand corner with the seasonal deaths of normal epidemic influenza. And so you can see that quite a few people die from influenza every year. But during a pandemic, the number is much larger. Now, if we look down, we can see that the virus was first isolated in 1933. So that's after the Spanish flu. But the other thing I want to really draw your attention to is that the first antibacterial drugs and antibiotics were not introduced until after that pandemic. Neither was the vaccine, which was introduced in the early 1940s. So there weren't an awful lot of tools to use when the Spanish flu was around. Remember that I said some of the disease associated with influenza can be bacterial co-infections. So the absence of antibiotics is actually quite important. And obviously the absence of a vaccine is crucial. And then you can see that mass vaccination really didn't come into effect until considerably later. What I've also highlighted on here, and I'll highlight it even more on the next slide, is Australia's role in influenza, and I got to do this. I can't help it. Australia was instrumental in the introduction of the discovery and the introduction of the first truly effective class of influenza antivirals, and that was the neuraminidase inhibitors. And they were discovered and developed from basically the early 1980s with the elucidation of the crystal structure of the neuraminidase through to their approval in 1999. So around about 55 years between isolation of the virus and approval of the first truly effective drugs. Then if you travel to the right on this figure, you can see some of the other events culminating on the far right-hand side in a more recent antiviral that was approved, and I'll talk about that in a moment. So this is the progenitor, the first effective neuraminidase inhibitor, Repurposing. Made a bit of a splash when it was first launched and then disappeared rather abruptly thereafter. It's an inhaled product, twice daily for 5 days. It really works. I've used it myself. Actually used it on my daughter as well. It definitely works without question. And it was discovered locally by a number of people contributing. Peter Coleman in the center there on the left-hand side, Wenyang Wu and Mark von Itzstein at the bottom there, who were the chemists involved, and Graham Laver at the top, who was involved in isolating neuraminidase, amongst others. Made the front page, and quite a splash. Now, suffice it to say, it is a member of a class of antivirals that work. But there's something worth dwelling on here, and that is testing an antiviral for a disease which is generally benign and which most people recover is actually quite complex. So the clinical trials in general use tend to show an improvement in days to alleviation of symptoms. Most people are going to recover anyway. What happens with the use of these antivirals, be it zanamivir, be it oseltamivir or whatever, is that they recover a bit quicker. And a lot of people interpret that as to mean ho-hum, you know, so what? You're going to get better anyway, so why bother? And this slide illustrates what happens when someone has severe disease. So this was an example of a patient that was hospitalized with severe influenza. And you can see at the top all of the various interventions that were used to try and rescue this patient, and had very little effect, including nebulized zanamivir, hydrocortisone, oseltamivir, etc., until they tried IV zanamivir with a cortisone. And you can see that there was a rapid recovery of lung function and a reduction in viral RNA. And I think this represents, it's a good representation. I know this anecdotal because it's a single case, but it's a good representation of some of the clinical data that's emerged since the approval of these agents. And that is, in a nutshell, the more serious the disease, the better they work. And that shouldn't be a surprise, really, because the more serious the disease, the bigger the impact, and therefore the bigger benefit one can demonstrate. Now, currently, for influenza, these are all the antivirals that are available. And you can see down the left-hand side here, the ones I mentioned, oseltamivir, zanamivir, the earlier ones, amantadine and rimantadine at the very top, they tend not to be used. They target an ion channel resistance emerges very quickly and only work for influenza A, and generally not recommended for use, but they were one of the progenitor antivirals. Peramivir is also a neuraminidase inhibitor, tends not to be used significantly. It's only approved for intravenous use. Lanionamivir is the long-acting neuraminidase inhibitor that I referred to in that earlier figure. It was worked on by Biota and Daiichi Sankyo and is approved and used widely in Japan. In fact, it's the largest selling neuraminidase inhibitor in Japan. Interestingly, it's a once-only neuraminidase inhibitor, so you only have to take it once, which is very interesting and useful. But it is inhaled. If we look across to the right-hand side of this figure, you can see there's some assumptions that are made, which I think I'd like to highlight in subsequent slides. So, for instance, if we look at zanamivir, it says likely to make up a minor component of stockpiling for pandemic use, for instance, because it's inhaled and it has limited experience because it's not sold very much. It's kind of a self-fulfilling prophecy. You don't use it very much, therefore you don't get much experience, therefore you don't recommend it for stockpiles, therefore you don't use it very much. And then it no longer exists. And that's exactly what happened. Real shame, because that compound has an excellent resistance profile compared to Tamiflu. Nice to say that lanionamivir has the same resistance profile. Not approved anywhere else outside of Japan. It probably won't be, but nonetheless is available in Japan. The two at the bottom, favipiravir, is unlikely to get widely used. It is teratogenic, but we'll come back to that in a moment because that's some potential uses for COVID. And then there's a really interesting endonuclease inhibitor that was approved only a few years ago, right at the bottom here, that also has some use in stockpiling and so forth, and also is a single dose agent. Now, let's talk a little bit about the impact of things on the use of drugs and the way drugs are used and how that can impact the way those drugs are used in a pandemic, and then the way the use or the lack of use of those drugs has an impact on disease and so forth. So a few things that they seem obvious when you start talking about them, but they're not obvious when you first start thinking about them. So if you're going to use a drug in a pandemic situation, it's actually quite important that the medical practitioners are used to using it outside of a pandemic. Because if they don't have a lot of experience in using it, they're not used to prescribing it, they don't know necessarily about the side effect profile without looking it up and so forth, drug-drug interactions and you name it. Part of this also hinges on perceptions in the medical community regarding the utility of antivirals. So if the perception is that antivirals are not very effective, they're not going to be the first thing they reach for in a disease situation. And that also impacts the way they're used, and then that impacts their availability in things like pharmacies. So, if they're not getting prescribed very often, they're not available in pharmacies. If they're not available in pharmacies, they don't get prescribed very often, self-fulfilling prophecy. And that then feeds into availability. So if the drugs are not particularly available in a non-pandemic period, it's very hard for practitioners to get experience. And if the drugs aren't available during a pandemic, obviously they can't be used. And if they're not in the supply chain, it's not easy to get them available during a pandemic. And then guidance is actually very important. Guidance to the medical community in the non-pandemic period and then during the pandemic. And then making sure that's communicated very effectively. So I wanted to give a couple of case studies. So one is Japan. Japan during the 2009 H1N1 pandemic. And this is the pandemic that, if you recall from that earlier figure, killed around about 200,000 people worldwide. Now, Japan had a decade of widespread use of neuraminidase inhibitors. It became a routine, if you like. If someone turned up at the doctor with influenza, it was routine for a diagnostic to be performed and a neuraminidase inhibitor to be prescribed. So there wasn't necessarily any stigma associated with that use. It was generally considered to be a good thing. So during the pandemic in Japan, almost 100% of hospitalized children had been treated with a neuraminidase inhibitor. And the next point is really important. Most had received their antiviral within 48 or 24 hours of symptoms. And these compounds work much better when they're given early. So what that really means, effectively, is they were receiving those antivirals in the community before they got sick enough to report to hospital. And then some of them got worse and worse and ended up in hospital. Of those that ended up in hospital, only 1% required ventilation, and there was only one death. Similarly, in pregnant women, there was a very high incidence of pregnant women giving neuraminidase inhibitors very early in the onset of symptoms, and no deaths were recorded. Now let's contrast that with a country that didn't use neuraminidase inhibitors very significantly, and that's the UK. Australia would be analogous to this. So neuraminidase inhibitors were not available in the UK. They were available in the sense that they were approved, but they were not available to use unless influenza exceeded a particular level in the community. Effectively, that meant they were not available. So a doctor could prescribe them, but you weren't going to get them because they're not there. And that essentially meant that doctors weren't used to using them. Pharmacies weren't used to stocking them. And there was a generally low acceptance of their utility. People didn't think they were very useful. In fact, when neuraminidase inhibitors were first approved in the UK, the chief medical officer actually said, "We don't want these drugs. We don't want people with influenza turning up at our surgeries." What happened during the pandemic? Only roughly 1 in 10 pregnant women and children were given drugs when they got influenza in the community within 48 hours of onset. A lot of them got drugs once they'd arrived at hospital, but that was too late. It was long, the period where they could have worked was generally long gone. And 12 women died and 70 children died as a consequence. Now, I know these numbers aren't perfect, we're comparing 2 countries, but you talk to Japanese doctors, they're absolutely convinced that they rescued a lot of their population because they use these drugs properly. Now, there's been a lot of work on 2nd generation compounds. This is one that Biota did, so I'm a little bit proud of this one. But this is a dimeric form of zanamivir that we were working on with the NIH and Daiichi Sankyo. It's arguably one of the most potent antivirals ever described. It's 100 times more potent than what was a very potent compound, zanamivir. CS8958 is LANI namivir. So more potent than that as well, and effective once after a single administration in animal models. This is a good example of a compound that was looking fabulous. It was partnered with GSK, was doing great, got through preclinical and completed at least 1 week inhaled toxicology, and the preliminary CMC had been completed, but it was not taken forward for commercial reasons. And that's an example of a compound that would have had great utility, but is never going to see the light of day. And this is just a few pictures of how it works. It basically seems to clump viruses together. You can see on the left-hand side here, electron micrographs of viruses that have been treated with this compound, and the viruses are all aggregated together. And we think probably through linking the neuraminidase heads on the surface of the virus, and then those electron micrographs on the bottom of the screen show these great big mats of virus on the surface of cells that have been linked together by dimers. And you'd love to think that that would present a really juicy target for the immune system. But anyway, that's an aside. So moving from influenza to HIV. So what we have with HIV, HIV as a pandemic emerged in 1981. Interestingly, it was identified because gay men in San Francisco were coming down with a lung infection that they really shouldn't have been coming down with. So they're coming down with pneumocystis, which is generally benign in the average person. But these young men were dying or becoming very ill with pneumonia. And the reason was not recognized immediately, but the reason was that they had HIV and their immune systems were completely compromised. So this otherwise benign bacteria was taking over the lungs. As you know, it's caused by a blood-borne retrovirus. There is no vaccine, despite a huge amount of work to get one. But there are many antivirals, and I want to talk about something which I think HIV brought in, which is the concept of combination therapy. And we don't talk about it a lot anymore because people seem to think HIV is taken care of. But you can see from these figures, which are 2019 figures, so they're pretty current, there's a lot of people living with HIV, and there's still a lot of people that die as a consequence of HIV. And this is a timeline, just to give you an idea, between the times that the virus was first identified in 81 through to the time that we had truly effective antivirals. And I want to talk a little bit about this because to me this is really cool and really shows pharmacology and pharmacy in action. And that is the concept of combinations. So prior to this, a lot of people said no one would ever be able to do combination therapy. It's just too hard. You're going to combine drugs that have different mechanism of actions, like different pharmacokinetics, different side effects, different drug-drug interactions, and so forth. Nonetheless, that proved not to be the case. and started off with a complex scenario where people, such as the gentleman on the right there, had to take complex sets of drugs in single dose form. Nonetheless, it did work, but there were big issues with compliance and so forth, because not because people didn't want to do it, because it's easy to get messed up. Through to the fixed dose combinations, which are shown by a representative in the bottom here, where you start combining multiple agents into a single fixed dose combination that's taken, in this particular case, once daily. This particular fixed dose combination is actually on the World Health Organization list of essential medicines, which is not straightforward to get onto, and not what one would have, as someone who was working in the HIV field back in the day would have expected. And so it's very pleasing to see. But when these drugs were launched, this is the first, what's shown here is the first launch of the heart therapy, the highly active antiretroviral therapy, back in 95. And you can see the deaths just plummeted. Also, the monthly spending changed as well to the better. And at the bottom, you can see that life expectancy then in these patients rapidly grew, such that nowadays they have the same life expectancy as the normal person. When I say normal, I mean someone who's not infected with HIV. I didn't mean that to be derogatory anyway. Neither did I mean that life expectancy is not necessarily associated with other adverse consequences, because it's not exactly the same, but the antivirals have really transformed the situation for people living with HIV. And it probably won't have escaped your notice that several of the drugs that are used in these combinations are nucleosides and nucleotides. So this particular combination is the combination that I had on the previous slide. And you can see at the top there, that's a non-nucleoside polymerase inhibitor, then two nucleoside polymerase inhibitors. I want to dwell on these for a moment because it's relevant for where we're going in a moment. One is a nucleoside, it's a non-phosphorylated nuke, and the one at the bottom is a nucleotide. That particular one is an acyclic nucleotide. You can see that it's phosphorylated, albeit with a non-cleavable linker, and also is a prodrug. And the reason I've shown two versions of it here, the one on the left and the one on the right, is it's representative of the way these compounds can have broad spectrum activity. So tenofovir on the left is used in HIV combinations, and tenofovir in the prodrug form on the right is an hepatitis B drug, but also used in HIV, various HIV fixed dose combinations. And then this has led to a plethora of drugs. So here's all the drugs currently approved by the FDA, for instance, for HIV. And you can see that an awful lot of them have the initials FDC alongside them for fixed dose combinations. So the great deal of fixed dose combinations have been approved. And what this does is it gives people living with HIV and their healthcare providers a lot of options as to switching their combinations should they develop resistance, should they become intolerant of a particular combination, and so forth. Okay, moving swiftly along to the current pandemic. So I think everyone knows it's called SARS-CoV-2, also the disease COVID-19. It emerged in late 2019 in China as a pneumonia of unknown cause, and then rather swiftly moved outside of China. You can see that the WHO announced a public health emergency in late January, and then a pandemic in early March. So things moved. For those of us who were sleeping through this, none of us were, of course, but those of us who may not have noticed the timeline, it moved fast. The virus itself has had a lot of speculation as to its origin. The data strongly suggests its origin is bat, possibly with an intermediate host. There is speculation that it emerged from, engineered in a laboratory. I would love to meet the virologist that could have engineered that. It's very, very unlikely based on the sequence. Importantly, it's an RNA virus, so it's a relatively mutable virus. But very importantly, it has no evidence of prior human immunity, apart from some small evidence that existing coronavirus may have some T-cell immunity that has been ascribed in a few studies. And there are no vaccines and no antivirals. So what does that mean? It means the population is completely naive, which is why it's moved so fast. And we've moved, at least from my perspective, very fast to counter it. There have been an awful lot of antivirals and an awful lot of vaccine approaches. And this is a generic picture of how a virus grows in a cell. And all the little red symbols there are points of intervention. Lots of different places you can intervene. And I would say almost all of those have been looked at or are being looked at for SARS-CoV-2. Then there are other things we can do, such as social distancing and wearing masks. I'm not seeing a lot of social distancing in these two figures. The top one is from 1918, and the bottom one is today. The point being that people in 1918 didn't have a lot of options from a medical perspective to intervene with that virus, but they could wear a mask, and they did. We're doing the same today. And I think that's entirely appropriate. It actually dismays me that mask wearing has become a political item. But anyway, let's move on. So what's happened? Well, I think we probably characterize it as an absolutely massive R&D response. Billions upon billions of dollars have been thrown at this. But I think it behooves us to actually think very clearly about the level of scrutiny us as scientists are under at the moment. We've never been under this level of scrutiny before. And it's scrutiny at the level of the media, the public, and electronic transmission of those outputs from the public and the media, which we've never had before. When you combine that with preprints and other releases of data, things can get out of hand very fast indeed. So back in June, it seems a long time ago when I say back in June, according to the European Pharmaceutical Review, there were 2,000 clinical trials underway. That's a lot of clinical trials. 398 of them were for two agents that I would say have little to no chance of working. 22 were on remdesivir. We're going to talk about that a bit more. And there were a lot of companies involved. And then there were a whole bunch of other agents being tested as well. Now, I've purposefully chosen a report from the New York Times of only a matter of days ago, because this is sort of the general public perception of what's going on. According to the New York Times, there are two interventions that are available today that are widely used. None of them are drugs. Two of the interventions that are promising are remdesivir and dexamethasone, and they have been subject to clinical trials, although they need more work. And then there's 11 tentative ones that have good potential for working or possibly could work, depending on the way you look at it. And then ones that are not promising because the trials have not been effective. And you can see that hydroxychloroquine and chloroquine are listed there, despite what a certain president would say. And then fraud and pseudoscience. This is something we really have to guard against, the fraud and pseudoscience. I'm just going to talk briefly about this, but I'd really encourage everyone to be incredibly vigilant about this. So this is an example of fraud and pseudoscience. Take silver. Colloidal silver will cure coronavirus. It will also get you thrown in prison. This is an example of somebody in Missouri who's a televangelist who was selling silver solution for people to take. And this is what happens if you take it. This is actually a condition, it's well known that taking colloidal silver, ingesting colloidal silver over a period of time will cause your skin to go blue. And it's permanent. Not a good idea, no benefit. This particular gentleman died in his 60s. Hard to know whether he died in his 60s because of ingestion of silver, but there is evidence that prolonged ingestion of colloidal silver can have organ problems and so forth as well. But it's not giving you any benefit, so why are you doing it? You know, you have to ask. And I really do have to ask my my host this evening what he has to do with this. As you can see, there's a certain effect of colloidal silver called the Tyndall effect on this. It's a joke. It means that it measures the dispersion of light in silver solutions. But I thought Ned would get some fun out of that. And then there's the whole concept of repurposing. So repurposing, there's a reason for it, right? Which is repurposing existing drugs. You can move fast. Potentially you're taking a drug which already we have the safety knowledge. And it involves genuine science, it can involve fraud, and it can involve politics, fear, and need. And I wanted to take an example here, and I'm not pointing any fingers, but I think it really is quite a cautionary tale. So this is some work that was published by colleagues at Monash. Absolutely nothing wrong with this work at all. Showing that the FDA-approved drug ivermectin, which is used as an antiparasitic widely, quite an inexpensive drug, inhibits SARS-CoV-2 in vitro. Excellent, nothing wrong with that at all, except the concentration that is necessary to inhibit SARS-CoV-2 in vitro vastly, vastly, vastly exceeds what is achievable in vivo. So here's a modeling study, for instance, that was published by Craig Rayner and colleagues, showing that the concentration that's achievable, even using 3 times the highest dose one can use in humans, is still orders of magnitude below the 50% effective concentration. So virtually no chance of working in this particular modality. There may be other ways of using it, perhaps in combination, perhaps in different dosing modalities that would be worth investigating. But what happened as a consequence is there was this study published in the Lancet and the New England Journal of Medicine showing that ivermectin worked in people. And you can see on the left-hand side here a really startling effect in a Kaplan-Meier survival curve of what purported to be COVID patients that were on mechanical ventilation plus and minus ivermectin therapy. It was fraudulent, but nonetheless it was published and was subsequently retracted. But the damage was done. And what happened was a series of Latin American countries very quickly indeed, moved to distribute doses of ivermectin to their population and endorse the use of ivermectin. So we're talking about a very large number of doses, for instance, 350,000 doses distributed to people, for instance. This is not small potatoes. And some of these incidents involve the distribution of agricultural product, what is essentially sheep dip, for human use by local politicians and so forth. And this is an example of that. And you think, well, that can't happen here, right? In fact, it was considered to be a terrible thing, and still is considered a terrible thing by those in the know in Latin America. But nonetheless, the fires are still trying to be put out. And the authorities have issued warnings about the use of this drug until it's been clinically proven. That being said, only a week ago, this appeared in our literature, if you want to call it literature. And one of our politicians is calling for the use of ivermectin to be used off-label. And I think this is a very good example of how even in a very, very well-informed society with extremely good science, these kind of messages can get completely out of hand. And so I think we need to be quite vigilant about how these things get reported and act as scientists whenever we can. Okay, so moving back to real science, nucleosides and nucleotides. As I mentioned before, they've got a long history as effective antivirals. They've got potential for broad spectrum. They're often not very straightforward to make, as those of you who have worked in the field will know. And from a biologist's perspective, testing them can be quite challenging. So for instance, if you have a nucleoside that's not readily phosphorylated, you have to set up assays targeting the polymerase with the phosphorylated form of the compound to ask questions about whether or not it works or not, and then think about using prodrugs and so forth. Okay, so let's talk a bit about remdesivir. Now, you've heard a lot about remdesivir, but it's important to emphasize that it's not yet approved anywhere around the globe, and it is investigational. It's an injected antiviral, likely to be useful for people in hospital. And it originated in antiviral programs around the HCV arena. And from a group of compounds known as C nucleosides that at the time were being tested by Gilead and others for broad spectrum activity. And it was also evaluated for Ebola. There's some limited evidence that it might have a survival advantage, but is under evaluation for SARS-CoV-2. And there seems to be more evidence there. This is the compound itself. As you can see, it's a phosphorylated adenosine derivative with a CN modification at the 1' position. And it's also a prodrug. And this, just to emphasize on the right-hand side, that it's an injectable. But I wanted to highlight, because I can't help it, a local connection, and that is the connection through to Biota. Many years ago at Biota, we were working on HCV agents in collaboration with Boehringer Ingelheim. And at the time, the compounds of interest in the field are shown on the left-hand side, one of which became particularly prominent in the field, the sofosbuvir at the bottom. And the compounds that we were working on the right-hand side here, which are basically C nucleosides, so that the glycosidic bond is actually a carbon-carbon bond. And the compound 1 in particular was particularly interesting. Now, at a similar time, others in the field were also working on similar molecules. If we look into the top right of this figure, we can see two molecules. Molecule number 3 was being worked on by a company called BioCryst. Interestingly, BioCryst also discovered the compound that Biota discovered in the previous slide, although we beat them to the patent. And then the compound number 4 was being worked on by Gilead, all using the C nucleoside platform. And then the compound on the bottom left-hand side, at least the SP isomer, is remdesivir, and you can see that's the prodrug phosphorylated version of compound number 4. I've put, we've got sofosbuvir and tenofovir there for comparison. And so I think you all know that what we mean by a prodrug is it has moieties on it that increase its likelihood of crossing a cellular membrane and masking the charge on the phosphate, such that one can deliver the nucleotide. And why would you want to do this? Well, nucleoside analogues are not typically readily phosphorylated. If you can bypass that very first phosphorylation event, they tend to move up to the triphosphate, which is the active species, quickly. But if you do not, if you require that first phosphorylation event, it means a lot of nucleosides are inactive. The compound that Biota was working on was very unusual in that it was readily phosphorylated as the nucleoside rather than the nucleotide. And this is just a comparison. So you can see this is remdesivir on the left-hand side. And for patriotic reasons, we have the Biota compound on the right-hand side. You can see the Biota compound was the 2'-C-methyl version at the sugar, but it had the same base. The next thing we were going to do after discovery of this particular compound was start modifying the sugars and attaching different sugars, which would have led us eventually, I think, to 1' modifications. But suffice it to say that didn't happen. You can see that the dog pharmacokinetics are very, very attractive there. With respect to remdesivir on the left, evidence of broad spectrum activity and human exposure, which is consistent with activity at the antiviral concentrations that you see in vitro, which I think is an important observation. Now, we're in the very early days of data acquisition for remdesivir. The data that's been acquired to date are positive, but there are some reports that I think are worth just dwelling on very, very slightly. So for instance, if we take the study that was published in the New England Journal on the top left-hand side here, you can see that people recovered quicker on remdesivir, 11 days compared to 15 days. And it says, but the effects on mortality were negligible. That resulted in similar comments on the right-hand side, for instance, to say that doesn't really cut the death rate. And I think the problem with it is it's a very small study. So we had mortality rates of 7.1% compared to 11.9%. There were only 1,000 patients in the study. So basically enough people didn't die, which is a horrible way of looking at it. But you know, reminiscent of what I was saying about influenza inhibitors back in the day, it's hard to demonstrate effects on things that don't happen very often in early clinical trials. So it's important not to dismiss a molecule because you don't get those effects in the early studies. Instead, look at everything else that's happening and form a view as to whether it looks like it's working, and then do further studies. And I think that's exactly what people have done, and which has resulted in approvals around the world, including the TGA, for instance, in the bottom right here, giving a provisional approval for the use of remdesivir in patients with severe COVID-19. Now, bear in mind, this is an injectable, so it's not going to be used in the general population anyway. It's going to be used in patients that are in hospital and very ill, which is exactly where it should be used. Okay, so just a few words to finish up. So 1918 and now. Back in 1918, there weren't a lot of options. This was one of the options that was put forward, was straight whiskey. And according to an awful lot of the reports, a lot of people are increasing their imbibing as a consequence, as many of my colleagues on the present call seem to have been doing before we started the talk, hopefully because they're all hoping to create an antiviral effect as we go. And as I mentioned before, the use of face masks and so forth was widespread in 1918. Note that not only are the children here wearing face masks, but so is the cat. And you think, my goodness, that couldn't happen nowadays, but you would be quite wrong. This is a picture of a cat wearing a face mask. I couldn't help it, had to show it. And of course, other things don't change. This is our friend arguing that the use of face masks was inappropriate compared to the situation in 1918 in San Francisco, where the police decided they would shoot a person for not wearing their face masks. It appears that we're slightly more tolerant nowadays, but I don't know how far I would push it with those gentlemen there. And then there are other thoughts on face masks that I won't go into, but suffice it to say that some of our politicians might need to rethink their position. And I'd just like to end with another Oscar Wilde quote, because I think it's rather relevant to the situation we find ourselves in. Not only the fact that we need to do good clinical trials, but also the fact that we're all undergoing personal trials at the moment. So thank you very much.
Thank you very much, Simon. Yes. No, and look, it is interesting that we're all undergoing these personal trials, and I do hope I don't end up looking like that. I thought he looked like Papa Smurf, the guy with the. But I think I might turn a different colour if I keep drinking this. But thanks, Simon. I think it was really informative. I think there's certainly many questions that people will probably have. I certainly do. But has anyone online got a question that they would like to ask? Please unmute yourself and well, start your video and please ask away. We've got a few minutes. I think we can engage in some Q&A. My first question, if no one else has one right away, has that I can't remember the name, but BTA, the biota compound, was that tested against Ebola and SARS and other viruses? Does that have activity, broader spectrum activity?
Yeah, the code is B1133.
And it's seared
of course.
into my
how could I ever forget? How could I ever forget?
It was something I very much wanted to do. Wanted to take one of the things we wanted to do was to take a platform of nucleosides and nucleotides that we developed and test them against a broad spectrum of viruses of serious emergent possibility, such as yellow fever and Ebola and so forth. The funding was just never there to do it, but it would have been something that I would love to have done. It was actually exactly what Gilead did. They assembled a group of a library of compounds, which just so happened to contain the precursor of remdesivir, and it was how they discovered that it had activity against a broad sway of viruses.
I guess my subsequent question, if we don't have others, is so what is the best approach? Do you develop one compound versus several potential pandemics, or do you develop several compounds against the ones that you think might be the most likely pandemics? What is your suggestion?
Yeah, it's very difficult. I think the answer is both. So if we take influenza as an example, it's likely that an influenza pandemic is going to result from a virus with all of the genes, or at least the gene functions, that are present in influenza today, in epidemic influenza. So it's likely that a neuraminidase inhibitor, to take an example, will work. Now, whether it will work all the time, whether resistance will develop, different matter. But you can at least say, well, that class is going to work for that particular high-risk pandemic pathogen. And then develop as many of them as possible in different variants and different approaches as well. Then there's the emergent virus that you don't know anything about. you didn't predict, didn't expect. And some people are going to say, okay, look, SARS gave us a heads up, right? SARS, MERS, this was coming. We didn't do a lot of work to find SARS, MERS antivirals because it's not around all the time. And coronavirus is common, causes the common cold. There's no reason to develop an antiviral. It's very hard to do the clinical trials. There's no market and so forth. So, in those circumstances, I think broad spectrum is the way to go. Identifying agents that you think are going to work across a wide variety of different pathogens. And then if you have the time, when it emerges, selective can be follow that. But how do you make a selective agent for something which you don't know has emerged yet?
Questions? Anybody else?
Yeah, Simon.
The big question I've got really is about the whiskey hypothesis. And I mean, Craig Morton, fielder hopping, is whiskey the answer to this? Because I'd love to know that. But no, look, you mentioned the importance of combinations, obviously, with HIV, and you showed with the flu, like there's at least 2 different targets there. Xofluza is a different target from neuraminidase. I mean, has any work been done on combinations, for treating flu?
It's emerging, not yet clinically, but it's emerging. It's a great idea. No reason why it can't be done. And actually no reason why one wouldn't be able to incorporate one of the early M2 inhibitors like rimantadine, for instance. Yeah. Because if you put it in a combination, the resistance issue disappears.
Could you say a few words about PVPI, Simon?
Jim knows that I've been working with a local company recently. So there's also a very, very simple approach, which is basically disinfection. Not antiviral, but disinfection. And the idea being to eradicate, at least transiently, virus in the area which comes into proximity with others. So for instance, if you go into surgery, one of the issues is MRSA emerging generally from your nostrils and causing illness. And that can be inactivated by disinfection of the nasal passages with povidone iodine gels. And the idea that a local company called Fibric is working on is disinfection of the nasal passage with a nasal spray for respiratory viruses. And that has really caught on around the world. There's clinical trials happening in Europe, in the US, etc. at the moment. Unfortunately, using homemade versions of PVPI, which I don't think is a good idea, because there could be issues with stability and side effects and so forth. But nonetheless, the idea being to provide healthcare workers and frontline people with something to immediately protect their nasal passages and arguably prevent immediate transmission from infected people that they might come in close proximity with, for instance. It's an interesting idea. Thanks, Jim.
Yeah, there is actually a result from a clinical trial in Malaysia. It's very small, but they were using throat gargle as well.
That's right.
And I think out of 5 patients treated, it's a very limited phase 2. They compared gargling with PVPI, a standard marketed variety you can get off the shelf. And 5 out of 5 patients cleared the virus within 6 days versus controls, gargling with water, 0 out of 5. So there's a number of different trials that are kind of following up on that at the moment. But it's something you never hear about. I like to think of it as POVID for COVID, but that's...
Oh, that is terrible. There are similar data on bacterial infections.
Yeah.
I'm not trying to trivialize it. I think it will work.
So Simon Sepp here. Do you think there's a silver lining to what's happening in Victoria? I mean, in no other state in Australia could you do a clinical trial at the moment, could you? There's not enough patients there. Is that a problem for us developing drugs here?
Yeah, you hate to say things like this, Seb, but the absence of, you'll get me on my hobby horse here in a minute. The absence of patients is a serious problem. Warehousing those patients for trials or therapies which might not have much utility is a really serious problem, because you can only use them once.
Yeah.
You know, so I'll take the example of the recently approved Russian vaccine, for instance. That's going to be, if they really intend to widely use it, as they've said, that will take cohorts of people out of the clinical trial environment, because you can't use them twice. And we will not have a placebo control. We will never know if it works if what they say they're going to do, they do. So.
Okay, thanks.
Hi, Simon. I have a bit of a maybe more left field question. I was just wondering, you know, with a lot of, as you mentioned, pandemics coming sort of from a combination from an animal source, is there something about certain animals that make them more, the virus more easily transmissible from bats, pigs, birds?
It's a really, really good question. I think it's probably something more about humans and their interactions with the animals. But that being said, you don't see a lot of examples of transmission outside of the mammalian kingdom, for instance. But I think the big driver here is humans impinging upon animal systems. You know, how often would people go into bat caves, for instance, in the past? Probably not very often. You know, would they expose a bat to a civet? Probably not. But that can happen now. So I think it's us interacting with their environments which is really creating it. But I think we want to be really, really, really careful about finger pointing. You know, we leap to finger point and say, okay, it's wet markets. Let's do away with wet markets. That's the end of wet markets. Okay, we'll close Victoria Market then. You know, stop saying stuff like that. Let's have a think about, you know, what is it that drives that interaction in the first place, and maybe get to that rather than trying to put a blanket ban on things, which is not going to work anyway, by the way.
Another animal question. I know animal models are pretty challenging with viruses, like for, you know, efficacy, like comparative efficacy studies for humans. Are there any animal models for coronavirus that are useful? Yeah, there are. There's okay, so let's go from the small animal to the large animal. Large animal, that's the non-human primates, which are very useful, thought to be predictive, but difficult to do and limited availability for obvious reasons. Moving back, the next animal model, arguably, that looks really good is the ferret. That's being, if we want to bring it home, there's a lot of that work going on at Geelong at the moment. And then there's the ACE2 modified mouse model. So mice that express the COVID receptor. The issue is getting them. The colonies were run down and not widely used, and then all of a sudden they went, oh Lord, I think we might need a few thousand of these, and so rather rapidly increase them. So yeah, there are animal models. There are. It's interesting. Thank you.
Hi Simon. Thanks very much for a great, really interesting talk. I just wanted to ask, do you think we should have been able to predict that coronavirus was a candidate to cause a pandemic? And could we have done anything after SARS in 2003? Could we have done anything better to make sure that we were more prepared for now?
What a question. Yeah, I think SARS gave us a huge wake-up call, and we didn't wake up. We got close to a vaccine and then stopped funding it. But I don't think it's fair to compare the level of funding that we have now to the level of funding that even might have been available for a SARS vaccine. You know, it's just order of magnitude is different. Personally, I think it would have been good to have run at least one of those vaccines to the ground and shown, just like the Ebola vaccine, that it was doable. But it would have required quite concerted effort on the part of funders, in other words, governments, to put many billions of dollars into something which may never have been useful. So politically, a really tough decision. The other one that I wish we'd done more of was broad spectrum antivirals. Yeah, but again, the funding was just not there.
Do you think this might make it politically more palatable to fund science better?
Oh, what an interesting question. I actually am deeply concerned that the opposite may be the truth. I'm deeply concerned that the COVID pandemic is causing people to leave science in droves. You know, the lack of funding to the universities, the cuts in staff. You know, one of my companies, for instance, Jumpstart, one of our lead scientists, he said to me, "Simon, I'm leaving science. I can't see a career." And you think, well, what a terrible situation that is. In the midst of something which requires science, somebody that's an excellent scientist is saying, that's it for me. I don't see any way forward. And I think there's a lot of people in that situation. In fact, I don't know about you, but I've encountered a very weird phenomena where members of the public say to me, "Oh, you must be doing really well at the moment because, you know, COVID, all the money that's being pumped into science." It's actually quite the opposite. Quite the opposite. Yeah, so that was a real downer answer to your question, Jeff. Sorry about that.
That's right. Oh, well.
I'm sure there's plenty of jobs in marketing, and I think Jim, you've got the running there for head marketer for iodine. But look, are there any other questions? We're heading on to 9:30. I know bedtime beckons, but I might just wind it up there. I will thank you again, Simon. It's been a fantastic talk, and I just want to make sure everyone understands this has been recorded, so we'll put this on the website and make that available to everyone for reviewing, and I think unless there are some pressing questions, we might wind it up there. And please, Simon, don't leave your house because there is a bottle of wine coming.
At some point. No, it's very kind of you. Thank you.
But I couldn't present it to you tonight, but that is thank you very much for tonight. And I don't know what to do. Maybe everyone unmute and we'll give it a round of applause.
Thank you.
Thanks.
Thank you, everyone. We haven't organised our speaker for next time, but please stay tuned, and we will email you. And otherwise, look at our website. The RACI website should also have notification of when the next event is. But on that, thank you very much, and we'll see you next time. Thank you. Bye.
Yep.